This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1119, recorded on May 31st, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 76 degrees Fahrenheit, which is 24 Celsius, and there's not a cloud in the sky. It's really beautiful. It's just about the most perfect weather you can have.
I am not in the U.S. I am in Stockholm, the home of the Nobel Prizes. I had dinner last night with Gunilla. I forgot her last name, but she's the chair of the Nobel Committee on Physiology or Medicine, the committee that reviews all the submissions. She said they're working on it now. But I think it's like Stockholm, 20 degrees C and partly cloudy. You know, the cool thing about Europe is that they use Celsius, centigrade, and nobody complains about it. It's really cool. Also joining us today from Austin, Texas, Rich Condit.
Hi, everybody. I am happy to report that we have had some recent relief from both drought and an early spring in the form of some really pretty cool storms that have been blowing through. Some people have read about this in the news. We have not locally had the baseball-size hail and that sort of problem or the tornadoes. We've just had some cool storms, and that's going to give us a little bit of relief, at least for our drought situation. And right now, it is 84 degrees Fahrenheit, which is just fine with me.
Also joining us from Montreal, Canada, Angela Mingarelli.
Hello, it's wonderful to be back. I feel like I haven't been here in almost 2 months. I've missed you guys. It's the same temperature as it is in Ann Arbor. It is also 24 C 76 F. And beautiful, as you can see outside my window. It is very sunny. There's lots of squirrels running around these trees and on my balcony. And yeah, excited for the show today to be back with you guys. I've been studying for my comprehensive exam. It has been very intense.
Wow,
it is in 3 weeks.
Good luck.
What does your comprehensive exam cover? Everything, since it's comprehensive?
Well, so it's everything adjacent to my project, which is... I guess I do like T cells and myeloid cells, so it can be anything in immunology, but surrounding myeloid biology, T cells, things like that. Also some aspects of mechanosensing, which we're starting to do now, which is a whole new field, which is cool. So learning new things about how cells sense their environment.
Excellent.
Mm-hmm.
Here it is 9:14 p.m., so I'm 6 hours ahead, and it's light. And it's going to be light another 2 hours. So the sun, it'll get dark at about 11. And then the sun rises at 2 a.m.
Oh my gosh, 3 hours of darkness.
The other night I woke up at 3 a.m. and it was bright. I said, oh, is it already 6 a.m.? And it's 3 a.m. It's really weird. And then, of course, in the winter, the sun sets at noon. It's dark all the time. Weird, right?
That would be hard to take.
Yeah, you get used to it.
When are you there until, Vincent?
Well, I am here in Stockholm till Sunday, and then I'm going to Finland for a coronavirus meeting. So I'm doing a podcast there.
You'll have even more light, right? More daylight hours.
Yeah, and it's north of here. It's 3 hours north of Helsinki.
So do they have any darkness now, actually? They might. It might be almost.
I don't know. It's a good question.
I spent, this was almost 60 years ago. I spent a summer a little north of in the neighborhood of Umeå in Sweden, which is way the hell up there. And as I recall, we got something kind of like a long sunset between 11 and 1-ish. Okay, it never got completely dark.
Yeah, well, the hotels have blackout curtains. You have to use.
Oh, okay,
right?
Sure, makes sense.
Otherwise, you wake up. All right, let's see what we have here for announcements. All right, the 2024 International Hepatitis B Virus Meeting. Registration and abstract submission is open. This meeting will take place September 11 through 15, Chicago, Illinois. And the website is hbvmeeting.org for more information. And Michelson Prize, Next Generation Grant awards of $150,000 to support early career investigators working to advance human immunology, vaccine, and immunotherapy research for major global diseases. These are awarded by the Michelson Medical Research Foundation, and applications are accepted until June 9. So get it in if you want to get these prizes. And what do we have for ASV, Kathy?
Well, in the continuing series of updates about special events and workshops, these are all advertised on the webpage under the area where the satellite symposia are, just under that. And then probably within a week, you'll get your program PDF and your mobile app links. And so you can check things out there as well. And so previously we highlighted the DEI one that's on Monday, and Think Like an Actor, Speak Like a Pro that's on Tuesday. And I want to tell you about the Tuesday and Wednesday Careers Beyond Academia workshops. These are right after dinner, and they're concurrent with scientific workshops. So depending on your scientific interests, you might not be able to go to one or both of these, but they are quite popular. And so we encourage you to think about going to those. Several of the other special workshops that we'll talk about in future TWiVs are about the education workshop, the communications workshop, and the assistant professor boot camp / workshop. And so you can check those out. They're virtually all free. They are all free once you're there at the meeting. So, and then somebody else wants to talk about the next part, and I will do the show and tell.
If you attended ASV in Athens or Madison last year or before, start locating your name badge pouch. ASV wants to be eco-conscious. Think sustainable. And if you can reuse yours, it helps the planet. They are black and look like this. Hey, Kathy's holding it up. Let me see. Remind your friends who are going to ASV but may not listen to TWiV. I wonder, I didn't go last year. Would I still have one from like the year before?
Did you go to Madison?
Familiar. I think so.
Yeah.
I mean, last year's the first TWiV, the first ASV I missed in like decades.
Right. Yeah. We made them generic. So you can see it's just the ASV logo, no year. And then down here is the meeting specific stuff that can be replaced every year.
So it's probably lying around somewhere.
Yep. When you run across it, you'll say, oh, that's where it is.
And I'll keep it.
Yes. Yeah. Good.
All right. Thank you, Kathy. On to our science. We have 2 Nature Communications papers today. That's just how it fell. And the first is entitled Longitudinal Viral Shedding and Antibody Response Characteristics of Men with Acute Infection of Monkeypox Virus, a Prospective Cohort Study. And this comes from Yang Yu Shen Yang Song Peng Zhu Guo Shen Liao Liu Zhang Kui Chen Chen Huang Wang Lu and Liu.
I think you did that all in one breath. That's really good.
Did.
Impressive.
Did.
That's like at least 2 measures, right, Kathy?
Exactly. Yeah.
Yeah, so it was challenging. And the affiliations at the end of the paper. So let's scroll down. There's a variety of institutions in China, as you might guess. Here we go. We have the Southern University of Science and Technology in Shenzhen. We have the Guangdong Laboratory for Diagnosis and Treatment of Emerging Infectious Diseases in Shenzhen. We have the Clinical Research Center for Infectious Disease in Shenzhen. We have the Provincial Key Laboratory of Tropical Diseases in the Southern Medical University. I think I said that? No. Southern Medical University. It's different. That is in Guangzhou, and the Hubei University of Arts and Science in Hubei, and the Bengbu Medical College in Anhui, China, and the authors Yang Yu Shen Yang and Song all contributed equally, and Young and 3 last authors are corresponding authors. Wow, a lot of shared stuff here.
Mm-mm. Mhm.
All right. Kathy, what's the summary here?
Okay, so as you probably guessed, they were investigating patients with the disease Mpox, and that's caused by the virus called monkeypox. Monkeypox virus is 5 syllables, and so we'll just abbreviate it MPXV, with the V at the end telling you that it's a virus. And that's only 4 syllables. And that way we don't have to say monkey, so it's either Mpox the disease or MPXV the virus. So also the title told you that the patients that they looked at were all men. 98% of them are men who have sex with men, and 2% are bisexual. And some of them were HIV positive. And so in this paper, they sampled many body sites and many environmental sites for viral DNA of the Mpox virus. Mpox disease-causing virus, the MPXV. And so they generate a huge set of data, and there's some things that emerge that are really helpful for thinking about non-pharmaceutical interventions when this disease breaks out, and mainly things about the environment, treatments, and possible vaccines. So I'll just tell you in brief the results, and then we'll see how much more in detail we want to get. But when they looked at the patients' body sites, the 100% positive sites were skin lesions, and the second and the third were the rectum and the oropharynx at 84 and 79%, so less than the 100% that you see for skin. The virus peaks in most sites 6 days after infection, except in the rectum, it's the highest levels of the viral DNA are right there at the onset, and then it declines. They examined serum from these patients for neutralizing antibodies against various MPXV proteins, and they identify a couple that seem to be really predominating in terms of being antigenic, and so those might be important for vaccine considerations. They didn't really find differences between the patients that were HIV positive and negative, except for the fact that HIV patients, the rectitis was associated with higher viral DNA levels in the rectum. Okay. And then the environment, oh my gosh, they look at so many different kinds of things in the environment. Pillows and toilet flush levers and doorknobs to the patient bathroom and all kinds of stuff. It boggles the mind, but the bottom line is they find the highest levels of virus in these fomites in the air conditioning vents, number 1, and the floor, number 2, and then cabinet near the patient's bed, it was number 3. And then over the 21 days when the patients were there, they didn't really see any change in the levels of the viral DNA in these various fomites.
Great.
So there you have it.
That's the whole
Wonderful
paper.
summary.
That's it. All right. Let's talk a little bit about MPXV, the virus. Rich, what do we need to know about this virus?
So it is a pox virus. The most notorious of the pox viruses is smallpox, which was endemic globally. Actually is probably grew up with humans, and as humans grew up and the planet became more densely populated with them, we ran into a big problem with this in the 16th, 17th, 18th centuries. If you lived in one of the urban population centers, like say Paris or London, you knew that you were going to get this disease, probably in childhood. So you knew as a parent that your children were going to get this disease. There was about a 30% chance that they would die from it. And it was absolutely the most horrific thing that you can imagine, with blisters that hurt and itched all over the body, including inside your mouth, and a bunch of attendant other symptoms like headache and nausea and etc. It was just absolutely gruesome. And in the late 1700s, there were various modalities of immunization, practices that had been practiced over time, but in the late 1700s, famously Edward Jenner did appropriate experiments and published the use of an animal pox virus that we need to do a new episode on as soon as I can get my act together. Okay. It's a related virus that comes from nature that we call vaccinia that cross-reacts with smallpox, but the disease is limited to a single lesion when it's introduced into the skin superficially. And the use of that vaccine resulted over the next, 150 years in the eradication of smallpox worldwide. So this is the family of pox viruses have a complex morphology. Most of them symptomatically present with lesions in the skin of pussy lesions of some sort or another. There are pox viruses of vertebrates and invertebrates throughout nature. The invertebrate pox viruses behave a little differently symptomatically. They kind of melt the larvae. Okay. Among these are the orthopox viruses in humans, which include vaccinia, which I've described, the vaccine virus, smallpox, monkeypox, I think camelpox is in there, and cowpox, and horsepox, a few others. These all cross-react with each other. So theoretically, any one of them could confer immunity to the other. Monkeypox is so called because it was observed in monkeys and transmitted from monkeys to humans. That discovery was made in the 1970s in West and Central Africa, and has been until very recently pretty much confined to West and Central Africa. But as many people are aware, sort of escaped on humans from Africa to Europe and the Western and United States and elsewhere. How long ago is that now? Like 2-3 years? In an outbreak that was ultimately controlled. And there have been periodic outbreaks in Africa. And this particular paper describes an outbreak in China. It seems to be transmitted by direct contact and requires pretty intimate contact. And so has been observed almost exclusively in populations of men who have sex with men or bisexual males.
Rich, I have a question.
Did I do too much?
I have a question for maybe some of the listeners are thinking of, we all hear pox viruses and at least my mind jumps to chicken pox. And then you talked about smallpox, but how is, if so, monkeypox related to chicken pox? Is there cross-reactivity? Are there cross-reactive antibodies? Is the vaccine for chicken pox potentially protective against monkeypox? Do we know? Can you give us the details?
No relationship whatsoever. Other than the fact that both of them cause blistering lesions, or poxes. And so they both historically were characterized by those lesions, and so they got called poxes. But actually, semi-ironically for this episode, chickenpox is a herpes virus.
Exactly. So different. I wanted to make it clear because a lot of people hear chickenpox and they think that, maybe my chickenpox vaccine is also going to be protective against monkeypox. But
and interestingly, during the smallpox eradication campaign, one of the major efforts was to recognize who had the disease so that you could figure out who to vaccinate and who you didn't have to vaccinate. And the biggest confusion was confusing it symptomatically with chickenpox. Okay. So you had to be educated to be able to make that distinction, because they are both characterized by blistery lesions. And there's a classic, you can look it up on the internet, the smallpox identification card.
Mhm.
And you'll see pictures of individuals with either smallpox or chickenpox side by side that clinicians could use to make the distinction. But otherwise, like I said, thank you, Angela, there's no relationship.
I mean, it can
So,
be confusing, right? Because the listeners
Oh,
hear
yeah,
pox,
yeah, of
pox, and you
course, of
think that
course.
it's related.
absolutely.
Yep.
Mm.
This recent outbreak started in May 2022, so 2 years ago.
Oh, and
And
we should say that, I'm sorry, we should say that there are two major clades of monkeypox in Africa, which is where it, this thing hangs out mostly probably in rodents
mm,
of one sort or another. And usually the normal situation is it's sporadic spillover from a rodent to a human. And uh, only rarely in that habitat in Central Africa transmitted human to human. The secondary attack rate is something like 10%. Okay, so it's mostly just periodic and sporadic spillover from a rodent reservoir or from a monkey that's been tangling with a rodent and
mm,
has picked up the disease to a human. Um, and the two clades, one of them has a case fatality rate in humans of about 1%, and one of them has a case fatality rate of about 10%. Big difference. Fortunately, the clade that has been spreading outside of Africa is the less pathogenic. So it has actually, in the European cases, I think the case fatality rate came out to be, uh, it's got to be less than 0.5%. I think it's
mm.
down by 0.1, 0.2%, less than even anticipated by the African data.
All right. So this outbreak began in May 22, and it appeared in China in September of that year. And the authors say that currently, since June 2023, the mpox virus has been endemic in several cities of mainland China. So what they wanted to do in this paper is find out, where is the virus on people? What are the kinetics of virus production? And where is it in the environment, to get more information about transmission and so forth? And then there's a small study on antibody conversion in these individuals. So the cohort of people, 139 laboratory confirmed mpox patients in Shenzhen, China. This is between June 23 and November 23. All of them are men. 77 out of the 81 hospitalized patients were enrolled in this study. They're about 30 years old. 5 of them had gotten smallpox vaccination during childhood. 72 of them men who have sex with men. The rest bisexual. 42 patients were HIV positive. Interesting. And we're going to see if there are differences between those and HIV negative people. Most common symptoms, skin lesion, 98% of them, influenza-like symptoms, fever, sore throat, myalgias, et cetera, et cetera. Most common location of the skin lesions, the trunk, extremities, genitals, face, perianal region, and at the bottom of the list, hands and feet. None of these received antiviral treatment. So they got genomes from all the 77 participants. They're all part of this C.1 lineage that Rich mentioned. They have 993 specimens from these patients that were collected, and these include oropharyngeal swabs, saliva, rectal swabs, skin lesions, urine, plasma samples, and then, as Kathy said, 1,633 environmental fomite swabs. This is from 49 patients. This is floor, call button, right? You're in a hospital, you have a call button. It's pretty dirty. Light switch swabs, television, remote control, bed handrails, et cetera, et cetera. Every place you can think of, even clothes, pillows, toilet flush handles, shower handle knobs, window handles, I suppose. And these were collected. They're stratified based on when they're collected. 1 to 7 days post DPO. What is DPO?
Onset.
Days post
Days
onset.
post onset.
Yeah.
8 to 14 days, and then 15 to 21 days. Okay, so there are a lot of numbers in this paper, and they give you like 10 numbers, and then they'll say those numbers correspond to this, right? Like over 100%, 88% showed positive detection of DNA in skin, rectal swab, saliva. So the top two are skin and rectal swab. Then we have saliva, oropharyngeal swab, and then plasma is at the bottom there. Only 2 plasma samples were positive for DNA, viral DNA, in the 15 to 21 day post-onset group. Most of them were earlier. And there's a big table in here. Highest viral loads are found in the skin, followed by rectal swabs, saliva, and oropharyngeal swabs. So that's skin is probably the major way it's contributed, followed by receptive anal sex. They looked at viral dynamics. When do you find the viruses in these different locations? So for skin, the viral loads peak at about day 6, and they decrease. For the rectal swab, high viral loads are found at the beginning of symptom onset, and then they decrease rapidly. For saliva and oropharyngeal, similar to skin, but viral loads are lower. And comparing HIV and non-HIV group, similar loads. However, for the HIV group, higher viral loads in the rectal swabs. Really very interesting there. Yeah, how about the environment? About half of these samples, 860 out of 1,633, were positive for MPX vDNA. And yeah, as Kathy said, the top ones are the air conditioning outlet, 69% were positive, pillow, floor, bedside cupboard. Let's see, included here, toilet flush handles, 45% were positive. So it's all over the place. It's amazing. So the virus is on skin, and the patients touch all sorts of things. It gets everywhere. So I don't know if I were the infection control person for a hospital, I'd have a room completely sanitized after an Mpox patient. I don't know, Rich, how long is this virus going to remain infectious in the environment? Do you have any idea?
This
I was
is
gonna
one of
ask.
the, first of all, we should say that they're only sampling DNA.
Right,
Okay.
right.
And that's a, I don't know if you want to call it a problem. It's a feature. They're of the study. They're not assessing virus. They talk about viral loads. That's not accurate. They're talking about DNA. Okay.
Yeah, that's right.
So that there's a big question mark there. Nevertheless, with respect to and we should also say that DNA is everywhere, right? It's really a problem.
Sure is.
So, but as far as stability goes, this is one of the toughest nuts to crack there is.
Mm-hmm.
This is an extraordinary. This thing was built to be stable, and in fact, even in the smallpox era, Kathy mentioned the word fomites. That means any inanimate surface, a pencil, a doorknob, a blanket that could be contaminated with virus and therefore transmit it to another individual. And bedding, clothing, etc. in the smallpox era was a real problem with this because a patient who used this stuff would leave virus all over it because it sheds out of all of these lesions and stuff. And it's extraordinarily stable and can be then transmitted to somebody else. So that's a problem. And, you're only looking at DNA on these sites, but the implication is that I suppose you could have DNA all over your body from, there was virus there at some point. Okay.
But are there other papers that have shown plaque assays done off fomites in like X amount of time, like afterwards trying to culture virus?
I couldn't quote you chapter and verse, but certainly there's been a lot of studies on the stability of of this virus or related viruses in on surfaces and stuff, and it is really tough. By the way, it's a double-stranded DNA virus with a unique capsid structure. It's unlike anything else, and it's almost, in my mind, as if it was specifically designed to hang around. I also didn't mention in the run up to this that monkeypox, smallpox vaccination should confer immunity to monkeypox, and that has been demonstrated in several cases. And in fact, the most recent generation smallpox vaccine, the MVA vaccine from Bavarian Nordic, was approved for use in immunization against monkeypox, mpox. I don't know. I'm getting a little confused about the terminology because I grew up with monkeypox. At any rate, so
well, the virus is always monkeypox
yeah,
virus.
Right.
The disease is mpox. Yeah.
right.
Right.
So, so that's why, as Vincent pointed out, there were a couple of individuals who had smallpox vaccinations as in their youth. So,
Which is also surprising because most of these people are around between 30 and 40 years old.
yeah,
Unless it's
the smallpox vaccination fell out of fashion, you might say, at about the time it was eradicated, which we're talking late 70s for most of the globe. When eradication was declared in 1978, the World Health Organization says, okay, not only are we rid of this, but at this point, when we're talking about first generation vaccines, which had certain hazards associated with them, the risk of dying from the vaccination is greater than the risk of dying from the disease. So don't vaccinate people routinely anymore. So that hasn't, people haven't been routinely vaccinated since the late 70s, and in this country, actually since the 60s.
Now, of course, the military is vaccinated, so it could be in
Military.
China. Maybe these were military people.
That's a
because
good.
the oldest person
that's a good
is
possibility.
51, I think, in the age range. It's 21 to 50 or 51, I think, from the table. So that's very young, 51, to have been vaccinated. No.
Yeah, but military would do it. Okay,
Military.
certainly in the U.S., you're going to have a population of military and healthcare workers that are vaccinated. They also said they weren't drug treated, that interests me because there are anti-pox viral drugs. One in particular developed in the U.S. called TPOX or tecovirimat. That's I would think could be available in China if there were appropriate arrangements made. So that sort of interests me.
So in terms of sorry, go ahead, Kathy. Go ahead.
No, I was just going to say, back to your question, Vincent, about like things you could do. To me, thinking about where they found the highest levels of the virus in the environment, seems like you ought to really be cleaning those air conditioning vents, and you really ought to be mopping the floor. Rich, would bleach be enough to do it?
Oh yeah.
Okay.
Bleach will do it.
Yeah.
They ethanol resistant? Do we know?
I know. Yes.
Yes, they are.
Okay. Because I have personal experience with that. Okay. Ethanol will not reproducibly do the trick. There's a great story that I'll tell you off camera sometime.
Yeah, I heard it when I visited. It's a good story. So there is a relationship between the DNA copy number and infectivity. So they mentioned a previous study that showed the lowest load of 6.6 times 10 log 10 copies per mil is the lowest where you would see infectious virus, right? So 4 million copies per mil, basically, that's the lowest you're going to get infectivity. So anything below 4 million copies per mil, you're not going to see infectivity. Anything above it, you will. So they say in our studies, the areas that had the highest proportion of swaps with loads higher than 4 times 10 to the 6 per mil were the deposition area, the bedside cover, and then the pillow. So they're saying you would likely find infectious virus there, but who knows if it would be enough to transmit. Right?
Well, to me, the big question, actually, there were two big questions in this, but I'll do the first one first, is, transmission. How are you going to get infected by this thing? And I would say, first of all, historically, that smallpox actually was transmitted as a respiratory infection.
Mm-hmm.
And you would inhale droplets or whatever, and it was pretty infectious. And it would replicate in the lungs, establish a viremia, that is, virus in the blood, which would then spread to other internal organs. You'd get amplification in those organs, and then a secondary viremia, during which it spread to the skin, and then you get the lesions on the skin. And man, you could have this thing cooking and not really feeling too much for at least a week. And it was often 2 weeks incubation period before you actually erupted it, into lesions. But the virus can also be transmitted by direct contact between lesions and, say, a break in the skin. And for non-smallpox orthopox viruses like vaccinia, or I believe monkeypox, predominantly, it is that mode of transmission that's important, really. Rubbing up against an active lesion, okay, with a portion of your skin that's either a mucous membrane or has some sort of microabrasion or even a cut in it. And I think it
Mm-hmm.
takes a significant amount of virus and quite intimate contact to make it happen, right? Fortunately, and that imposes a pretty strict limit on transmission. And I note in this paper that one of the things that they noted was that they couldn't find much, if any, DNA in blood.
Mm-hmm.
So that suggests to me that these individuals probably are not mounting a viremia, which would be consistent with everything else I know about monkeypox. It does raise the question of if people have lesions on multiple sites in their body, then how's that happening? And I think that's probably spread by contact as well, for the
Mm-hmm.
most part, rather than a viremia. I'm guessing here, but that it's an educated guess. And that's not uncommon with one of the things when you got a smallpox vaccination, which was a scratch of vaccinia virus in your arm that created this active blistery lesion. One of the things that you are or were, when that was the primary vaccine, cautioned was don't scratch that sucker, don't even touch it, and get rid of the Band-Aids. Now are fomites, right?
Mm-hmm.
Be careful as you manage them. Otherwise, if you scratch this thing or you're not careful with a Band-Aid and the virus that's on your finger from scratching it or on the Band-Aid makes contact with a mucous membrane or another cut, you're going to spread it around. And that happened. People would get it in their eyes or other parts of their body. And my guess is that in these individuals where you have multiple sites of infection, that's probably mostly what it is contact spread.
Okay. Next thing they do is look at antibodies against quite a few viral proteins, 1, 2, 3, 4, 5, 6 different viral proteins. 94% of the participants give antibodies, IgG antibodies against at least some of these. It depends on the protein, they're different percentages. But the interesting thing that I found was that they find neutralizing activity in 97% of the samples collected after 7 days. So it just takes 7 days for you to make neutralizing antibodies to this virus.
So I have a couple of comments about the
Yep.
immunology. I want to make the point that this is makes a stark contrast with what many people are now familiar with as through the SARS-CoV-2 epidemic. Okay, in that case, we were able to make a vaccine based on expression of a single protein.
Mm.
Although some of these antibodies will neutralize virus in an in vitro test, in order to get genuine immunization with this virus, there's no good single protein candidate that works. And all of the successful vaccines have been live attenuated vaccines. People have been working on making protein vaccines or messenger RNA vaccines, but those all consist of trying to figure out what the most prominent antigens are, antigens plural, and making vaccines that consist of several of these, like in particular the immunodominant ones that they study in this paper, the ones that you mount the best immunity to. So immunization against this thing is a much more complex problem than something like SARS-CoV-2.
So it would be finding the spike protein of Mpox would be very difficult, is what you're referring to. That
Yeah,
there is
there ain't
not
no
one that
spike
is
protein.
immune. Exactly. For the listeners,
There
that's
are
what we're referring to.
there are at least in the case of vaccinia, at least there are at least 4 different proteins required for or that seem to participate in the attachment of the virus to a cell. And then there's a complex of about, it's at least a dozen, I think maybe 20 proteins. That's the entry fusion complex for it to get in. So it's an extraordinarily complex process.
Wow.
Not only that, but I don't know what the correlates of protection are. I don't think it's entirely antibody, where, as usual, we're leaving out the whole T cell response,
I have to
right?
remember T cells, yes.
Yeah.
Which they don't really talk about they talk a little bit about the immune response, but only regarding like immune infiltration in the rectum, depending on if they're like because of the mucous membranes being inflamed and things like that. But they don't discuss at all T cells or immune.
It is
immune response,
impressive.
the
I think the
adaptive
most
response.
impressive thing about this paper, as we've already discussed, is that there's virus, or at least virus DNA, and probably virus everywhere. Okay, and it's not going away easy.
Yep. All right, so that's it. I just want to highlight a couple of things. So, for diagnosis, this is one aspect of the results that they swab all these places on people. What would be the best for diagnosis? Skin lesions would be the highest sensitivity. You find the most virus here, followed by rectal swabs and saliva. So if you don't have a lesion, but you suspect someone might be infected, you could do rectal swab or saliva to diagnose it. They also talk a lot about the high occurrence of genital and perianal lesions, supporting this idea that close contact during sex is a dominant form of transmission, which we knew before, and the rectal swabs have the highest viral loads, but that they decline rapidly after symptom onset, which doesn't make it the highest risk for transmission after those early days. The patients with HIV and rectitis have the higher peak viral load and higher DNA load and much higher DNA loads during disease progression than non-HIV patients. So they say there's high probability of sexual transmission, especially in men who have sex with men with HIV and rectitis. So those 2 factors are an important point there. They do talk about surface cleaning protocols. It's important for sanitizing and personal protective equipment to prevent onward transmission in healthcare settings, I suppose. Anything else?
Well, there again, what I think of is, there's virus everywhere, but if, and I believe it's correct, that the primary mode of transmission is getting a decent load of virus on your broken skin, then the probability of transmission from those is relatively, and I emphasize relatively low. It's not zero. Okay, you got virus everywhere. You gonna have to be careful. But I've on numerous occasions spilled virus on my skin and just cleaned it off, and it's okay.
Yeah.
But if you then spill it into a cut, it's not okay.
Yep. So one last comment about symptomatic versus asymptomatic infection. So with smallpox, we believe that symptomatic infections are the majority and that they play the biggest role in epidemic spread. The same was thought for MPXV infections, but now there's more recognition that asymptomatic MPXV infections can play a role in transmission. And they say this is probably because there's this dense sexual network of men who have sex with men. So it's sexually transmitted as opposed to perhaps casual contact, right? Where you're going to have, even if you may have lower viral DNA loads in a asymptomatically infected person, if you have that much more intimate contact, you're more likely to get transmission. So that's something that's really interesting, and that's was something that puzzled people about this outbreak because they'd never seen so much person-to-person transmission. But the virus got into these MSM sexual networks, and that was where there's very intimate contact and really facilitated transmission.
And importantly, it was, I think, mostly good information and education
Yeah,
that put the
Yeah.
lid on it. Okay,
That's
when
right.
people understood what was going on, you can control this by appropriate measures, behavioral measures, basically. And this paper contributes that to that in several ways, in that it really emphasizes where the virus is and where it's going to shed from and how it's transmitted, and that it's going to get everywhere in your environment as well. So that if it gets into your sexual network, you got to be careful.
Yep. Hard to be careful when you're having sex. That's the thing.
True.
It's hard not to have contact with
Right.
someone, right?
Can we
But
quote you
definitely
on that,
using
Vincent?
certain forms of protection will help,
Yeah.
limit
Yeah, but
transmission.
it helps.
Yeah.
But it's unless
True.
you sit across the room and wave to each other,
Yeah. Right.
it's hard to
Not
avoid.
not usually what happens.
Oh, you could wear a body condom, right? There's such things, I suppose, but then it's not very much fun, I suppose. All right. Their second paper is another DNA virus. How about that?
Isn't that great?
Two DNA viruses. So I am virus agnostic. It doesn't matter. Just viruses, as long as it's a virus. This is also Nature Communications. Gene editing for latent herpes simplex virus infection reduces viral load and shedding in vivo. I picked this because I've always been interested in whether we can handle latent herpes virus infections. The authors are Aubert, Hake, Strongen, Clauser, Loprieno, Stensland, Santo, Huang, Hirian, Stone, and Jerome. They're from the Fred Hutchinson Cancer Center and the University of Washington. Did anyone prepare a summary that they'd like to do?
I didn't, but if nobody else wants to go for it, I can do it. I've been thinking
Go
about
ahead.
this.
Go ahead.
Okay. So, herpes viruses, large family of viruses, once again, throughout nature, but each species has its own set of viruses. Among humans, there are 7 or 8 different herpes viruses, I think. There's herpes simplex virus type 1 and 2, which I'll talk about a little more. There's Epstein-Barr virus, which people are familiar with as causing mononucleosis. There's cytomegalovirus, and there are HHV 6 and 7, which cause rashes of one sort or another. I don't know if I got them all, but that gives you
VZV,
an
man,
what?
VZV, VZV.
Oh, VZV. Sorry, chickenpox. Yeah, we already mentioned that. Varicella zoster, chickenpox, and shingles. These are fairly large double-stranded DNA viruses that have a protein capsid surrounded by then a lipid envelope as well. And they all have, they're genetically related, but they all have in common the phenomenon of establishing a latent infection. All right. This manifests in different ways because different subgroups of herpes viruses establish their latency in different tissues. Okay. Like it could be lymphoid tissue in the case of EBV, it's B cells, or in the case of the herpes simplex viruses, it's nervous tissue. The two that we are most familiar with are HSV simplex viruses, are HSV1, herpes simplex virus 1, and HSV2. Herpes simplex virus 1 causes cold sores. herpes simplex virus 2 causes genital sores, and they are more or less geographically constrained in that fashion. And the deal here is that you get a primary infection. So let's talk about HSV1 of an epithelium, usually a mucous epithelium like your mouth or your lips or something like that, and you get this blistering lesion that's a cold sore. The virus is replicating, you're shedding virus from that lesion. And then what happens is that the virus travels up, in the case of these ones, the simplex viruses that establish latency in neuronal tissue, they travel up the axons of these neurons into the nuclei in ganglia in your head and neck, and the genomes reside there pretty quiescent, not doing much of anything in terms of either gene expression or genome replication. And they reside there indefinitely. Herpes is forever, we say. And occasionally they can reactivate, usually under situations of immunologic stress of one sort or another, and generate new virus, which then travels back down the neurons to the tissues that they innervate, which will be the tissues that you started with, and create now a new blistering lesion. I think, in fact, what's going on is there's probably a certain level of activity going on all the time in the nuclei of these things, but your immune system keeps it tamped down. And when your immune system is compromised to any significant degree, like called cold sores, when you got a cold or something like that, or UV light will do it, or stress will do it. So if you're out sunbathing instead of studying for your exam, okay, you're a candidate.
And then you might start worrying because of your exam, and you wouldn't be able to
Exactly.
distinguish it's the stress or the sun. But,
Exactly. And so that's why you get these recurring lesions. They'll show up and then go away, show up and then go away. Interestingly, in the case of HSV1, the one that causes cold sores, there's well over 90% of the population that are infected with the virus, but only about a much smaller percentage, maybe 10 or 15%, actually show up with symptoms. And the same goes for all these other viruses. All of us, by adulthood, harbor latent genomes for basically the whole gamut of herpes viruses, but only a small percentage of us ever show up with symptoms. So, like HIV, and there are drugs that can be used to treat these. There are vaccines for some of them, like varicella zoster / shingles. There's a vaccine for that now. Vaccinology has been difficult for most of these, so there's not a vaccine for HSV1 or HSV2 or any of the others, as far as I know. But the problem, like HIV, is that even though there are some drugs that you can use to control the symptoms, or even, prevent them under most circumstances, as long as there's latency, you're not going to get rid of it. There's no cure. All right. So if you want to cure it, you got to get rid of those latent genomes that are hanging around in the neurons. And that's what this paper is trying to do.
So, Rich, I have two questions. One for the listeners, one for myself. For the listeners first, and just in general to clarify, and if we know, which I believe we do, what is the difference between chickenpox and the herpes zoster virus? Is it the same thing? Is it so with shingles and chickenpox? There are a lot of people don't understand the mechanism of how shingles appears, and is it a latent form of chickenpox? Why does it happen only with age? If you were vaccinated against chickenpox when you were younger or not? What's the information? What do we know?
Chickenpox and shingles or varicella zoster are the same thing. And this is another one of these herpes viruses that establishes latency in nervous tissue. Okay, so when you get infected with chickenpox, the virus establishes latency in nervous tissue, and a lot of these are around the abdomen, though some in the head and neck. And if you're lucky, it never shows up again. But in particular, in people as they get older and their immune systems the wheels are starting to fall off. Okay. This thing can reemerge, much the same as cold sores or whatever. And it's the same varicella zoster virus that you were infected with in chickenpox, and it reemerges as shingles. And interestingly, the pattern that you get on your skin is a reflection of the nerves where the latency was established. You get these unique, quadrants of your head, the lower left part of your face or the upper right part of your face, or one portion of one side of your rib cage. Extraordinarily painful. Thank God I haven't had to deal with that. So, in fact, there is a vaccine for chickenpox. It's a live attenuated vaccine. And the first shingles vaccine was simply a double dose of that same vaccine used in adults. Now there's a newer vaccine that is a protein vaccine. Herpes has some of these same problems that I was talking about with pox viruses, where the entry and et cetera, all the antigens that you might want to make antibodies to are much more complex than something like SARS-CoV-2. So it's taken a while to come up with a protein vaccine that was effective, but there's now a protein vaccine that's effective against shingles. And I don't for the life of me know why that isn't used. And I don't think it is for chickenpox now. Probably just we're lacking the trials so far.
Yeah, I think they're doing trials.
Yeah, that would be a preferable vaccine, I think.
Yeah, it's a great vaccine.
My other question that was for myself, that was a wonderful explanation. A question for myself would be, we talk about, this reactivation, the virus goes latent and there's a reactivation, and we all say it's because of immunosuppression, or it could be in an immunosuppressed state. But what does that mean? Have the immune mechanisms actually been studied to see what it is that is causing this reactivation? Because by saying immune senescence or immunocompromised, okay, what does that mean? Do we know what's actually happening within the cells? If there is has anyone done mechanistic work?
Yeah, for sure.
I don't know.
Yeah, I could tell you.
You do know? Okay, good.
Vincent knows.
So in the latent state, the viral genome is in the nucleus of neurons and ganglia. It's silenced. Only a non-coding RNA called latency-associated transcript is made. The silencing is because the DNA is chromatinized, it's wrapped around histones, and the histone is compacted by methylation. So it's transcriptionally inactivated. When you get stressed, a neuronal protein kinase is activated. It phosphorylates the histones and releases the repression, the epigenetic repression. And now you get all three classes of viral transcripts, immediate, early, and late, made all together. The entire transcriptional program of the virus proceeds, and then the viral proteins are made, and you get genome reproduction and the production of new particles. So it's an amazing mechanism.
That's so cool.
Stress does this thing with the neuronal kinase, and it reverses the epigenetic silencing. How cool
so that's
that?
Much more fundamental
cool.
than immune compromise.
Yes. Now, many people in the field say that was figured out in mice, and so who knows what it has to do with people, but it is really a beautiful mechanism, right? There's a phospho attached to one of the proteins, and it just lifts the methyl repression, and then gene products are made that further remove those methyls, and then you get the full activation. I think it's a beautiful model. I love it.
That's so cool. Yeah, I was discussing it. I was reading the paper this morning, and in my lab with my another PhD student, Dinesh, and I was like, what are the mechanisms? People always say, latency and activation. I didn't actually go into to look into it. I was like, I'm sure Rich, Kathy, or Vincent will know. I will ask them. So that's very, very cool.
Yeah.
The one other thing I want to just point out, this will come out, but in introduction to this paper, just to make the point, is that animal models for herpes virus infections are really tough.
Yeah.
Okay.
Yeah.
And it's really hard to model this stuff in animals, and that makes all of this research so much more difficult. But that'll be obvious.
So mice are easily infected with herpes viruses. they establish latent infections, but they don't spontaneously reactivate unless you do something. So you have to put them on skis and put them on the top of a mountain, and maybe they would reactivate. But they don't do it on. That's one of the problems.
Which is
So in this
interesting
paper,
because you can
Yeah.
stress them, because you were just talking about stress
yeah, of
induced, right?
course.
So I guess it's
Oh,
not
you should
the same
you should
signal.
see what people have done over the years to stress these mice. Oh my God. They put them in hot water baths. They put them in a 50 ml conical and dip them in a hot water bath to heat stress. Oh my God.
That's terrible.
You don't want to hear about it. It's terrible. But we'll see some examples of that today. So basically in this paper, the antivirals will reduce your symptoms, but they don't get rid of the viral DNA. We're going to use gene editing to try and remove that viral, to inactivate that viral DNA and see if that makes a difference. This is going to be done in mice, but you need to do something before you get into people. So what they do here is they use a mouse model where they infect the mice. They can either add virus intravaginally or in the eye, intraocular infection, and the virus establishes latency. And then they can ask, if we deliver a meganuclease to these cells, can we cut out can we inactivate the viral DNA essentially? Now, a meganuclease, think of a restriction enzyme with a really long recognition sequence. So restriction enzymes typically recognize 4 or 6 or 8 or slightly more bases. These are really longer. So you could tailor the nuclease. It's called mega because it recognizes a big sequence. You could tailor it to your virus. So they have meganucleases that target specific herpes simplex virus genes, and they deliver them to mice using AAV vectors, which we've talked about many times here. And
But Vincent, what is a nuclease? So we know what a meganuclease is, but what does a nuclease do?
Sure. Well, a nuclease is a protein with enzymatic activity that cuts DNA. Right? That's what a nuclease is. And so they're saying, let's cut the herpes DNA in nerve cells, which should inactivate infectivity. And let's see how efficiently we can do this and whether we reduce shedding. So they put this meganuclease in an adeno-associated virus vector. They tried lots of different vectors, but basically in this mouse model where they have the eye infection, if they do this experiment, so they infect the mice, then they infect later, like some days later, 30 days later, with the AAV carrying the meganuclease gene, and then they wait some more times, like 30 or 40 days, and then they take out the ganglia. So up there near the eye, it's the what is it?
Trigeminal ganglia, or
Dorsal root ganglia?
Superior cervical ganglia.
Yes, superior cervical
the
ganglia.
SCG, true.
SCG, and they could just grind it up and ask, do we see a reduction? And so with these AAV vectors, they see 48 and 41% reduction in HSV load, which is cool that you're getting. This is working. The meganuclease is cutting the viral DNA, so there's less infectivity. There's less viral DNA when you assay it. And they say, well, what if we combine more than one meganuclease that targets multiple viral genes? So they try that. They have 2 different viral gene targets, and that gives you really good further reduction in viral HSV loads in these ganglia, right? They do the same thing with genital infections. They do intravaginal infection, let it establish. They give a AAV meganuclease injection, and they get 98% reduction in HSV viral load in the dorsal root ganglia. I presume they're doing PCR. Is that correct?
I presume.
I believe so, yes.
So they get really good reductions of viral load in the ganglia. Okay, but that's not the whole story. You want to reduce shedding, right? When you reactivate a latent infection, you make infectious virus and it's shed. We'd like to reduce that as well. But mice don't spontaneously reactivate, right? So you have to do something. And they do something I've never heard of before. They use an inhibitor called JQ1, which has been shown before to reactivate latent HSV in neuronal cultures in cells in culture. This JQ1 is a bromodomain inhibitor. basically, bromodomains are in many proteins. They're amino acid sequences. They're also in histones. So I suspect that this JQ1 is unwinding the histone in some way and turning on transcription, which is going to give you viral reactivation, much like the kinase I told you about does. So they don't know if it's going to work in mice. So they said, does JQ1 work? So they infect mice, and then they give them JQ1 intraperitoneally. And so these are latently infected mice. You get shedding in the eyes. These are the ocular model in 56%, 5 of 9 animals. If you don't treat them, 0 of 9 animals shed virus. If you just let them sit there and then you sample the eye, they don't shed virus. So this works. It's not 100%, but it's working. By the way, they say here, a direct comparison suggested that JQ1 may be a more powerful reactivation stimulus than hyperthermic stress, which is what I was telling you. They put the mice in a water bath, right? It doesn't work as well. And they give
And
sequential
definitely
treatments,
better
which
for the mice. Sorry.
yeah, also for the
Definitely.
investigator who wants to stick mice in water baths.
Exactly.
Right?
Nobody wants to do those experiments.
So they can do multiple treatments, and you get repeated shedding episodes. It's random. The shedding is random. It's never all mice. Sometimes it's 10 out of 12. Sometimes it's 1 out of 12 after 3 treatments. It's really random, unfortunately. But you can, and they actually talk about this. They can never get enough mice to make a statistically strong case in any one experiment, right? Because you don't.
This problem with it being sporadic,
Yeah.
stochastic is the word that they use. I've seen dealt with in other herpes papers by taking a group of animals and over time plotting a cumulative lesion score or a cumulative shedding score. Okay, and it so that you know over time, as more and more animals shed virus or score positive, the curve goes up, and it's a nice way to visualize the whole thing.
So actually, they do something like that at the end here. They combine all their
That's
data, right?
They have a meta-analysis.
Meta-analysis. Okay, so now they have a nice model where they can establish a latent infection in mice, and then they add this JQ1, and a fraction of the mice will start to shed virus. So they say, okay, does our meganuclease inhibit that? So you take latently infected mice, you treat them with an AAV encoding a meganuclease, 2 different meganucleases targeting 2 viral genes. Then a month later, you give them JQ1 and you swab their eyes every day for 4 days. So they find that treated mice have a reduction in viral loads in the ganglia. If you take the ganglia out, again, it confirms what they found before. But after JQ1, 3 of 10 of the dual meganuclease treated mice had detectable virus in eye swabs compared to 5 out of 10 in the control animals. So, only half of them made virus after the JQ1 treatment, 5 out of 10. And then the meganuclease brings it to 3 out of 10. It's not great numbers, but it's an effect, right? And the copy number goes down. It goes down. But I don't need to tell you the numbers. They measure the DNA copies per mil, and the meganuclease brings it down. 95% reduction, basically, in viral shedding. So it seems like their model works.
So they had 10 mice, right?
Yeah.
We said 10, I think.
No, here they had here they had 10, yes, and then they repeated it, and they had 8 mice, right? So in the 10, there was a 95% reduction. And they repeated it with 8 mice, and they got 100% decrease in total virus shed. So even though you only get a few animals making virus after JQ1 treatment, you can substantially reduce their shedding with this with the meganucleases. All right, well now, this sounds great, but what about side effects of the vectors, of the AAV meganuclease? People have seen before that AAV, giving people a lot of AAV, like 10 to the 14 viral genomes per kilogram. This is mice. It's a lot of viral genomes. You get toxicity. And so they've also seen toxicity in their experiments. These animals are getting 3 times 10 to the 12th viral genomes per animal. They have liver toxicity. They have some
I think weight
neurologic,
loss and
weight
overall
loss.
decline, general decline.
Yeah.
They
So they
said.
say this is not a good sign. Let's try and address that. So they say, well, what if we give less numbers of viral genomes? And so when you do that, you have fewer side effects. You see fewer foci in the liver. They still see a reduction in virus shedding that seems to be acceptable. Um, but these animals still have evidence of liver damage. They still have evidence for neuronal injury. And so they say no mice showed detectable signs of neuropathy. Yeah, but they can't talk to you and tell you they have a splitting headache or something, right? So they're still worried.
Hmm.
So they also evaluate this, by the way, in the vaginally infected mice. They also see a reduction in latent genomes in the dorsal root ganglia, and they also show that JQ1 induces virus shedding in the vaginal model, and that the meganucleases can reduce the viral shedding. Now they do their meta meta-analysis. They combine all their data because no one experiment is terribly convincing. So they have 171 swabs from AAV meganuclease treated animals. They have 99 swabs from control animals. They say there's a trend toward a substantial reduction in viral shedding after meganuclease treatment, and it's statistically significant. The probability of shedding is decreased with the dose of the AAV and the duration of meganuclease therapy. So, I mean, the numbers look good. It looks like this is trending in the right direction. And finally, they go back to the toxicity issue in a couple of ways that are interesting. So, so far we've talked about 3 serotypes of AAV with 2 different meganucleases, right? So you need to use a different AAV serotype for each dose. So that's 6 unique vectors. So it's complicated. And if we wanted to do this in people, oh my gosh, that would be such a regulatory nightmare. So can we simplify it? So they use a different meganuclease that recognizes a sequence in a viral gene that's duplicated. Okay, so they say we just need to go in with one meganuclease here. Will that work? They say good reduction in viral loads in the ganglia, and it works almost as well as the combination. And there's no liver inflammation. There's low in liver inflammation. There's no neurotoxicity. It also inhibits shedding in the JQ1 latency model, a 90% decrease in shedding. So using this single meganuclease seems to help as well. And finally, they say, well, these AAV vectors can infect many tissues. They can infect neural tissues, which is what you want because that's where the genomes are, but they can also infect liver. What if we use a virus that only infects neural cells? And they exist. So that would be, in theory, that vector would no longer be able to infect liver cells. So they have such a vector, and they show that in treating mice with high doses of that does not lead to hepatotoxicity. They do have neurotoxicity, but they can decrease the viral loads to address the inoculum to address that. And these neurotropic vectors, we'll call them, have also decreases of ganglionic viral loads, 68 to 70%, and decreases in shedding. So I like that, you have a proof of concept that you could cut the viral DNA and reduce shedding, but you can also reduce toxicity and side effects because you need to do that to get this in people. Now, this mouse experiment is not going to be the only thing you do before you go into people, but you probably have to do non-human primates, but you have to do something to minimize the toxicity. So I like this because it's something, the only way you're going to get rid of herpes viruses, right? You have to cut
Well, the same. I'm sorry, Kathy. Go ahead.
I was just going to ask, are non-human primates a good animal model for herpes viruses?
When I said that, I thought, I don't know if this is true.
I can think of herpes virus Cymeri, but that's not a human herpes virus. So.
They do. I don't remember where it was, but they mentioned using some other animal. Oh, they talk about guinea pigs, not non-human primates, right?
Guinea pigs, rabbits,
right?
People use. but now you're getting into bigger animals, and I don't know about the non-human primate thing, but mice, guinea pigs, and rabbits are
Yeah,
the most
that's what I
familiar
heard.
to
Not
me.
yeah, you're right. So, I mean, the point here is that you can design an AAV delivered meganuclease. It can get in neurons and it can edit herpes genomes, right? Because the reduction in the infectivity in the shedding is because you're cutting the viral DNA. So I think that's pretty cool.
Yeah, to me
That's
it's
really cool.
proof of concept, as you said.
Mhm.
Okay, with a really long road to go, but not an applicable to other. I mean, I think of HIV. HIV, same problem. Is getting
Yep.
rid of the
Yep.
latent genomes.
They also point out that, um, this may affect transmission. That's the big deal here, right? If you can reduce shedding sufficiently, you're not going to transmit, and that's of course how we spread it so efficiently, right? So parents of children give it to them in their saliva. They kiss their kids and they give them herpes viruses. So if you could somehow reduce the shedding, you might be able to interfere with that. And there's some, we didn't go over this, but there's some serious problems associated with herpes virus infections, not just cold sores, but it can be much worse. And so this is a big problem.
No kissing.
Yeah, right. Good
Don't
luck with
kiss your
that.
babies.
Good luck. Good luck.
Or
And
your
also,
camels.
or your camels.
Or
By
your
the way,
camels.
so this is all done with herpes simplex 1, and they say these meganucleases are not going to work for type 2. So we have to design new ones, but we're working on it. Stay tuned.
Yeah.
I think that's a cool paper. I
Mhm.
like that. All right, let's see. What do we? It's 10:30. I want to do.
10:30.
Well, it's 10:30 here.
We're in
It's what
different
is it?
time zones.
And it's still light there, right? It's just as light as it is for Angela.
It's pretty light. Yeah, it's pretty light. I want to do at least one email because Angela's here, and there's one for her. Okay, so if you go all the way to the bottom of the email, it says "Save for Angela." I've saved this for weeks now, Angela, for you.
Okay, yes, because I haven't been
So
here. Wait,
why don't you read it? It's from Ezra.
let's see. Yes. Ezra writes, "Hello, TWiV team. I come to you with a veterinary question. I recently learned that the rabies vaccine for dogs is not approved for use in other canines. From the American Veterinary Medical Association, data on the pathogenesis of rabies in canine hybrids are insufficient to develop evidence-based quarantine policies for these animals. Consequently, public health officials may not permit 10-day post-bite quarantine of canine hybrids after they bite a person and may instead require euthanasia and rabies testing. Likewise, public health officials may require euthanasia of canine hybrids as they are exposed to a rabid or potentially rabid animal, regardless of whether they have been previously vaccinated with a rabies vaccine labeled for domestic dogs. I'm curious if there's evidence that wolves and dogs have differences in their immune systems and or in how rabies infection affects them. Ultimately, dogs, wolf dogs, and wolves are all of the same species. It seems counterintuitive to me that a vaccine shown to work in dogs would not work in wolf dogs. At the same time, I understand that rabies is not a disease to take risks with, so without specific research into wolf dogs, we shouldn't just assume the vaccine will be equally effective. Do you have any thoughts or insights? Thank you, Ezra." So, I think that, it could potentially work, but how much research is going into wolves and on a wolf immune system? And it still is, a wolf isn't a dog. And I added a paper here where there was this publication in, where was it published? In Animal Genetics. It's called Comparative Analysis of the Blood Transcriptomes Between Dogs and Wolves. And this paper was interesting just to show that they took blood from wolves and dogs, and that they saw that there were 524 differentially expressed genes, and then a lot of these were associated with immune function. So I think that that there are differences between wolves and dogs, definitely, and we can see this like just by this one publication showing that there's variance between immune genes involved with, I think some of them, they say that their results suggest that wolves might have greater resistance to pathogens, hypoxia endurance, and aerobic capacity, for example. But they also live in very different climates, or they live in different environments, right? And it's not to say that it wouldn't work for a wolf, or a wolf dog, but there's not enough research in that. And until we actually test that, we can't say. And because rabies is such a dangerous virus, it's also not worth not euthanizing the animal in case of an exposure, or a lot of the guidelines as well. Like if you look on the Merck Veterinary Manual, which I've spoken about other times, they also speak about how it would only be used, a rabies vaccine would only be used in the case of a endangered species or a species that is in like in a conservation program or within like a zoo. I know now the word zoo is a little bit faux pas, but on a conservation program, or that is part of a breeding program or something like that. And then you would have to do titers afterwards, like antibody titers, to see if the animal is protected or not. But that's not going to be done in most cases. So to be safe, we euthanize animals, those that have been in contact with any other animal that could potentially have rabies. Maybe dogs or wolves, if we vaccinated them, maybe it would be protective, but it's unclear because we haven't done those studies. So
Okay.
So canine hybrids, is that just wolf dogs or are there other canine hybrids?
I think it's just wolf dogs. I actually went recently to British Columbia, and I went to, so in Canada, like West Coast Canada, and I went to a wolf dog sanctuary, and it was really cool, the wolf dogs there. They cross like huskies and malamutes with like wolves and things like that. And those are the only wolf dogs that I know of. I'm sure that actually maybe like jackals could maybe get crossed with like domestic dogs in certain areas of the world, or other like the dingo in Australia. Maybe there's like hybrid wolf dogs there. That's not a wolf, but I mean, it would be like a canine hybrid, like a canid hybrid. I'm sure there are others, but that's like the most commonly, at least in North America.
Have you ever read Never Cry Wolf by Farley Moat?
Have I? Did I read that in school? I'm going to say no, but like people.
So you're studying for your comprehensives, right?
Yeah.
This is a great distraction from studying for your comprehensive. I always used to do a lot of my reading when I was supposed to be studying for exams. This almost ought to be a pick of the week. This is an off-the-cuff pick of the week. Never Cry Wolf by Farley Moat. Read the book first, okay?
Okay.
It's a quick read, and it's great. And then go see the movie.
I've heard of it, definitely heard of it, but I've never.
Charles Martin Smith, man, does just an outrageous job on this. Great stuff.
It'll be good procrastination for reading.
Yep.
Let's do a couple more. Let's go back to the top. And Rich, can you take that first one?
Let me see here. Yeah. Bill writes, Dear TWiV gang, thank you for covering our HBV DMS paper on TWiV. You all did a great job highlighting the key points in an accessible way. HBV is an excellent virus to nerd out on for anyone interested in molecular biology. Thank you for putting it on people's radar. All the best. Bill Schneider, longtime TWiV fan. You're quite welcome, Bill. It was a terrific paper. Good fun. Was he like first author on the paper or something like that? Or no, he was one of the senior authors. Is that right?
Yeah. One of the senior authors.
Yeah, we really enjoyed that. It's good fun.
I love when the authors catch us, and it makes you understand that people are listening, right? I like it.
Sometimes we forget people listen.
Yep. Kathy, can you take the next one?
Yes. Adrian writes, Hello, TWiV. I'm a graduate student taking a virology course at Caltech by 115, and I had a question regarding the Lancet paper discussed in TWiV episode 1115 on May 19th. The paper was regarding a self-replicating mRNA vaccine, and my question is about the potential promiscuity of RNA-dependent RNA polymerases and the effects of vaccinating someone with a concomitant viral illness. For example, many vaccines are safe to receive while also sick with a cold or some other mild illness, including the current mRNA vaccines. Could the RDRP from a self-replicating mRNA vaccine be used by an already present RNA virus to replicate its genome? Would that potentially pose a risk to vaccinating individuals who are sick? Thank you, Adrian. So what comes to mind for me is the idea, at least from what we talked about previously, is this cis-acting activity of the RDRP. And while there's not evidence for that in a lot of the other RNA viruses, Vincent, I think you alluded to maybe it would be an explanation for something for the polioviruses, enteroviruses, and things like that.
Yeah.
So
I think there are also signals that
Yeah.
are specific on genomes for their polymerases, right? That wouldn't go across
Right.
virus families. Although, if you had, so let's say you got a self-replicating SARS-CoV-2 vaccine. They don't exist, but maybe we would have one. Then you got a common cold coronavirus, with the, but the incoming virus, the second the infection has got its own polymerase. So I'm not sure it would be any worse in the end, right?
Yeah.
Because the vaccine is probably going to be injected into your muscles. It's going to be relatively, maybe not. Maybe it's going to go to many different places. But I think the virus that's co-infecting has got its own polymerase. So I don't see why the self-replicating would be a problem if it could even recognize it.
But it's an interesting question.
Interesting
is.
question. I
It
agree.
is. It is. I had a discussion here today about mRNA vaccines and self-replicating RNA vaccines, which I thought was a great idea, right? You guys did it in my absence a couple of weeks ago. But they said that in general, we got lucky with coronaviruses. They don't seem to work well with other coronaviruses. But what really works well is, the Novavax vaccine worked really well in the few people who got it, because you start with so much protein to begin with. It's hard for the mRNA vaccines to catch up to that. And apparently a lot of the protein made of it from an mRNA vaccine is misfolded. It's not in the right place in the cell. So giving a bolus of protein, Shingrix is a great example, right? It's a glycoprotein. It's a great vaccine. And maybe if you had made an mRNA vaccine, it wouldn't be as good. Anyway, I hadn't heard this kind of discussion before. I thought it was really interesting. And one individual said, maybe we're not going to see any more mRNA vaccines. We just got lucky that this one got out really quickly in a pandemic situation, that's really good. But there's a professor here at the Karolinska. She said she was telling the press, wait for the protein vaccine. It's going to be great. And of course, it never didn't come until it was so late, right? So that is a good question. I like that. Yeah. I mean, I think that there are going to be issues with these self-replicating vaccines. I think people are going to be scared of them, frankly, but we'll see. All right, one more. This is another Caltech student. My name is Yi, and I'm a student from Caltech, currently taking Virology by 115. I have a question about COVID-19 self-amplifying mRNA vaccine you mentioned in episode 1115. Well, there isn't a COVID mRNA vaccine, even a candidate, but that's a theoretical thing, okay? Since self-assembly mRNA vaccine will encode RDRP for the spike protein mRNA, if a patient that just got infected by SARS-CoV-2 but doesn't have symptoms gets the injection of self-amplifying mRNA vaccine, will that cause the virus to spread even faster and lead to worse symptoms, okay? The way I'm thinking about it is that RDRP will increase the rate of the spike protein expression, and if the cell is already infected by SARS-CoV-2 and the expression of spike protein is the rate-limiting step, then faster spike protein production will lead to faster maturation of the virus and thus faster spreading. So it's a good question, right? It's a similar, except now we're using the same virus that you're being immunized against. So you get vaccinated with a self-amplifying COVID spike encoding mRNA vaccine, then you get infected with SARS-CoV-2. I don't know the answer, right? I mean, the two polymerases can exchange and work back and forth, right? Unless there's a cis-acting preference, as Kathy said. I don't know if that's the case. It's hard to know if that would be an issue or not. I seem to think that the vaccine remains localized, but many people have said it spreads systemically and gets to many other organs, right? So in theory, it could go to the lung or the upper respiratory tract. And I think you have to do some experiments, but to look at that, I'm not sure. You
Good
could do an
question.
animal
Yeah.
model, and you do vaccination and infection. You see if there's any untoward effects,
Right.
right?
That's the kind of question the safety wonks
It
are
is a
going to
good
want an
question.
answer to.
It is a good question. I like it. All right. Let's move on to some picks of the week. Let me scroll all the way down here. Angela, what do you have for us today?
Okay, so mine is actually from I asked Dinesh, someone in my lab, another PhD student. I was like, yo, do you have anything like really cool this week that you read about? Because I love to ask people in my lab, and a lot of the time they have great things. And he's like, oh, I saw this thing in the New York Times this morning. So it's called Unconventional Sex Let Anglerfish Conquer the Deep Ocean. And it was written by William Broad, and it's actually wild. So anglerfish, which a lot of people know, there's over 150 species of them. Not that people know that, but people know that anglerfish have like this little light on the front of their head and they live in the abyss. You know, some parts of the ocean are 7 miles deep. And obviously there's not a lot of light there. There's no light. So finding a mate, finding a female or a male, literally finding one another can be very difficult. Then this can limit the reproduction of anglerfish. So basically they explain all of this in this article, and they say that the males are actually parasites to the females. So basically, if a male finds a female, the females can be up to 3 feet long and the males are a few inches long. So they're tiny in comparison. And the males literally bite. So they latch on to the females with their teeth. And then they basically become a parasite and like a source of sperm for the female. And some of them latch on only for a short amount of time, but some of them stay there indefinitely. They lose all of their other appendages and literally just become testicles attached to the female, testes, I should say, which is wild. And then there can be multiple males attached to the female at once.
Oh my God.
This is amazing. And multiple. And there's a picture. So there's a picture of like a tiny little male and then like a huge female with like a little male attached to the side of it. And I thought that this was actually amazing. So the fact that anglerfish have this that multiple males are attached to the females, that they're so tiny and that literally the males disintegrate and just the testes are left attached to the female in some cases.
But
I
Angela,
thought was amazing.
We've always known that men are parasites.
I did not say that.
I know, I said it. I'm a guy. I said it. Yes. That's so funny.
Very
you would
cool.
imagine that having multiple males would be good genetically for that, right? You get a more diverse pool of sperm to get a more robust offspring. That's pretty neat.
But I never thought about it. Finding a male or a male finding a female in an abyss. Also, obviously there's pheromones involved and things like that. But in the bottom of the ocean where there's no light, there would have to be things like this. Because if not, they're not going to find another male. Is not going to find another female necessarily. So if they want to reproduce.
Angela, do you know how the males find the females?
I think it's via pheromones probably. And
they find them delightful. That's what Dixon would say.
That's a Dixon. I was going to say that's a Dixon.
There's a schistosomiasis where, in order for eggs to be made, so you're infected with a male and a female, and that's when you make eggs. So they get in separately, and I always said, Dixon, how do they find each other? He says, oh, they find each other delightful. It's pretty good. Same thing here, right? Okay. Yeah. Thank you, Angela. That's very cool. Kathy, what do you have for us?
Well, I have two things. One is Doonesbury from last Sunday, which at least 3 different sets of people sent me. Go check it out. And we shared it with the TWiV hosts. And it's a cartoon that's making fun of a candidate for president of the U.S. who's not one of the 2 major candidates. And he's... I heard something now that says he's not anti-vax. He just is sort of vax questioning, but we think he's actually anti-vax. And so it's him being kind of tormented by a couple of viruses in a nightmare situation. So you should check it out. And then the second thing is something that my lab manager sent me. It's a link to something called the Publish or Perish game. And it's not yet available, but I went ahead and paid the $1 so that I'm on the mailing list so that when it becomes available, I can get the game. But it's publish as many manuscripts as you can before they're scooped. And so it's a group game. It's one of these sort of more complicated ones, evidently, where there's, different things each player does in their turn. And players race to publish manuscripts with useless nonsense while sabotaging each other's research and delivering, quote, very helpful, quote, comments, reviewer 2 style.
It's amazing and terrible.
And I think it's, I think it's not necessarily just scientific academic publishing, or like one of the cards shows a practical field guide to unproductive meetings and organization time wastage. The zen of not finding your car in a parking lot, a journey into acceptance and despair. So anyway, it looks like it might be fun. You could click on this link and hook up with it and pay the $1 to, get advanced information when it becomes available. And they've got reviews of people who've played it and really enjoyed it. So I don't know, it just seemed kind of fun.
Cool. That's very cool.
That's cool.
Rich, what do you have for us?
I just ran across this somewhere. I'm not sure. It's actually a Facebook page that's bigger than just this pic. The page is called Sky, so it's an astronomy page. But this particular page within it is titled Giant Sunspot Region 3664 and Resulting Auroras. And it's just a bunch of photographs of the sunspot, this big thing that's been causing all these auroras, the sunspot itself, and a bunch of auroras. And I thought it was a really wonderful collection of these things.
Those pictures are gorgeous.
Yeah.
Wow.
There's been some great
Wow.
APOD pictures as well.
Yes.
Astronomy picture of the day, which is, if that is not your default page on your browser, it should be. Okay. There was a terrific video the other day, Kathy, of the sunspot. Just amazing stuff.
I'm on that page now because, the solar X flare as famous active region returns May 28th. I think that's the one with the video. Yes. So I have that link. I could put that in if you want.
Yeah, sure.
Yeah.
Wow.
All right. My pick is a article in Science. Mysterious sea urchin plague is spreading through the world's oceans. So they have, what sea urchins are, of course, and they're dying.
Of course.
There's a photograph here. Dead sea urchins washing ashore on Réunion Island in the Western Indian Ocean last year. There were tons of them all over the beach. So they were trying to figure out what's doing this, and it turns out it is a ciliate that is killing a variety of urchin species in the Gulf of Aqaba and beyond. It came from the Caribbean Sea, and it's never been known before to kill sea urchins. So it seems to be killing multiple species, which is concerning as well. So you can read this article and find out how they figured that out and what they want to do about it. They have a photo here of a dying sea urchin that kills them within days. So kind of interesting. What are you going to do about it? They say, well, there's not a lot we can do. And if it keeps going, we should before that get all these different species and put them in, tanks in captivity so that we can have them to re-release them when it's over. So
I wonder if this is going on in California. My first thought is for the sea otters, this is their favorite stuff.
Really?
Strongylocentris purpuratus.
Ah, that's interesting. By the way, this is written by Sean Cummings. What did you say? Which species of urchin?
Strongylocentris purpuratus.
Because there's lots of different species of sea urchins. There's like the long spines, there's short spines, and then there's like even purple ones. Like where I lived in the Canary Islands, we had just where I lived, we had like 10 different species, and some of them are considered beneficial and some of them are considered like parasitic and very detrimental for the ecosystem. A lot of them were very detrimental when we were surfing and would land on them, and then we would get all of the spines like in our shoulders and in our hands, and then they get infected and you need to like get them out with needles. Yeah, it's unfortunate.
The original paper is in Current Biology, which we can put a link to as well. Mass mortality of diadematoid sea urchins in the Red Sea and Western Indian Ocean. So this is a mass mortality event, MME. We have a listener pick from Abdul Aziz, biomedical scientist. Dr. Andrea Love answers pseudoscience questions from Twitter in an informative and enjoyable way. This is a YouTube link. Thank you, Abdul. That'll do it for TWiV 1119. Oh, I want to mention this article that I picked, this science article. I'm looking at it here. I'm in Stockholm, and all the ads are in Swedish. So it just shows they do local ad serving.
Hmm.
I have no idea what they're about, but yeah, that's interesting. All right. That is TWiV 1119. You can find the show notes at microbe.tv/TWiV. You can send questions, comments, pics to TWiV@microbe.tv. And if you enjoy our work on this and other science programs of Microbe TV, we'd love to have your financial support. You can go to microbe.tv/contribute. Our company, our corporation, microbe.tv or Microbe TV, is a 501c3 nonprofit, which means in the U.S., your contributions are federal tax deductible. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure thing. Always a good time.
Angela Mingarelli is at McGill University, Immune Vet on Twitter. Thanks, Angela.
Thank you. I had a great time. Everyone have a wonderful week.
You're going to be another 2 months before you come back?
No. Now, after my comprehensive exam, then I'm going to come on all the time.
All right. Well, good luck on that, okay?
Thank you.
I'm sure you'll do well.
Thank
I'm
you.
Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.